Thanks, thanks, Jesse. So again, to everybody that's joined us, thanks for joining the seminar this afternoon. You've joined because you know, of course, that it's going to be given by Wendy Harwood from the John Innes Centre. And, you know, I've known Wendy for a long number of years, and she just told me before she came on, or before you guys came on, that she's been at the John Innes Centre forever, which I guess I could say the same about the Scottish Crop Research Institute and now the James Hutton Institute. I've been here forever, so I know what it's like. So Wendy's led the transformation group, crop transformation group at the John Innes Centre for a number of years, and she's always been a great source of help and advice for us when our transformation stuff has crashed. And they've managed to sort out what the problem is and try and tell us how we can actually rectify it. So she's been a fantastic resource, probably for UK crop transformation, and still is, of course. And today, I think she's going to tell us a little bit more about some of the resources and some of the work that they've been doing on transformation in cereal crops, in barley, I hope, which I'm told is her favourite crop. So, Wendy, over to you.
Okay, thank you very much, Robbie. I'm hoping that you're all hearing me loud and clear, and if there's any problems, somebody will let me know. Yeah, I mean, it's a real pleasure to talk to the International Barley Hub, because, as Robbie said, actually barley has always sort of been my favourite crop. It's one we spent a lot of time working with, and we're very fond of it. And obviously talking about genome editing, which is such an exciting topic at the moment. So okay, so I'll get started and hope that the slide share and moving forward is all working. So okay, so genome editing in crops in general, in barley. Of course, we're using the CrisprCast system like most other people, and I'm sure you would have all heard multiple talks about using CrisprCast in genome editing in crops. So I'm going to not go into loads of background detail, but only to say that I think this system is, it's certainly exceeding my expectations of the way that it's going at the moment. And I think we're still only at the beginning. And of course, the beauty of the system is its relative simplicity, the fact that it only requires two components. So the first component being the nuclease, so often Cas9, and the second component being the guide RNA, which is guiding the nuclease to its target sequence. And at that location, the Cas9, the nuclease, will cut the DNA. And in its simplest form, if it's making a double strand cut, what happens next is that the plant repair machinery kicks in plants, normally a non-homologous end joining process. That is not precise, so it sometimes makes mistakes, and that mistake that's made at the cut site, which could be a single base pair insertion or deletion, a small deletion or a base switch, is often sufficient to knock out the function of the target gene. And that's the main way that we use this technology. So in thinking about, so my title talks about a toolbox, and I'm really pleased that I've got the opportunity to talk about this. So just to give you an idea of what I'm planning to do today. When we're thinking about genome editing, because the first thing that we've got to think about is we've got to deliver. It's okay, it's simple. We've only got two components, but we have got to deliver them into our crop. So I'm going to spend a little bit of time right at the beginning thinking about delivery. I'm then going to move on, and because the second thing that we all want is increased efficiency. So I'm then going to talk about the toolbox that Tom Lawrenson, who I'm hoping is in on the call and is going to be available later to answer some questions, that Tom has developed. So this is it's a Golden Gate based toolbox of components for genome editing in wheat and in barley. My main focus, what I'm going to be talking to you about today, is barley and showing some barley data, just a couple of bits of wheat just to show how things are working in wheat. So we want the high efficiency, but we also need those tools to enable us to do a lot more. So to do have a greater range of outcomes and more complex outcomes as well. So we'll focus a little bit on that. The toolbox actually is about 70 plasmids. don't worry, there's no way, I'm not going through 70 plasmids, that would be extremely tedious. So I'm just going to show a few examples of bits of data that Tom used to decide what was most efficient and what should be included in the toolbox. And then finally, of course, we've got this fantastic system, we have all these tools, but of course we want to see it used in the real world. We want to see outcomes, we want to see impact from this. And for that, the regulatory framework is really important. That's something that's been taking up a lot of my time recently, so I'll say a few words at the end about that. So let's get started. The delivery of the editing components, we're using a standard transformation system. It's an agrobacterium-based transformation system. We inoculate immature embryos with Agrobacterium tumefaciens. We select on hygromycin, regenerate plants through selection, and grow them up and transfer them to the glasshouse. So this system, with a few tweaks, we've used now for many, many years. It usually averages about 25% efficiency. So it's pretty good. But of course, if it was better, the better it is, then the more efficiently we can introduce our editing components. And the other thing with the system is this system was set up in the Spring Barley Golden Promise, which everyone works with because it's so good for transformation in tissue culture. But we want to be working in our genotype or a variety of choice. So it's really important that we improve our transformation system so that we've got a lot more choice and a lot more opportunities to work in the varieties that we want, rather than being limited to something like Golden Promise. So, okay, so there are actually two barriers to this perfect situation where we can transform whatever we like at high efficiency. The first barrier is if we're using Agrobacterium-mediated transformation, which we are doing all the time, is that the barrier is the susceptibility to Agrobacterium. And this is due to plant defense responses, and the fact that in some species and some genotypes have a more robust, I guess, response, if you like, so they're less susceptible to Agrobacterium. So if there's anything we can do to increase the susceptibility to Agrobacterium, then we can move, we can greatly increase that initial interaction. And just looking at these embryos here with some gust staining, move from something where there's very little to something where there's a lot. And there's different methods that have been used to do this. Years ago, using increasing the numbers of copies of virulence genes, for example. I'll just highlight one recent study, which I think has a lot of promise. And this was a study from Raman et al. It's published recently in Nature Communications. And what they did is they actually engineered Agrobacterium tumefaciens to express a type 3 secretion system from Pseudomonas and use that to deliver effectors which suppressed the host defense response. So, and this is quite an attractive approach because, using an engineered Agrobacterium strain, so to enhance transformation. And they used this over a range of crops, including wheat, getting increases in efficiency up to 400%. I've just included a little figure with the wheat data there. So this is an interesting opportunity, and it's to tackle the problems of transformation efficiency from this end, from that initial interaction with Agrobacterium end, as it hadn't been very much done in this area. I just want to show you one other thing when I'm talking about this, and this was a finding that Tom made and noticed as we were doing transformations. A lot of the transformations we've made a whole range of different knockout mutants in barley. And often we then go back and we transform those mutants either to make additional mutations or to put in additional genes. So, and we know with one particular mutant, this is an RLK4 mutant, when we transformed that, we immediately began to notice he looked really good. There's an awful lot of regeneration going on here. So these are wild type embryos transformed. You see regeneration beginning, and this is the RLK4 mutant. So RLK4, it's a receptor-like kinase. It's involved in the activation of plant defense responses. So it's quite likely to be affecting the susceptibility to Agrobacterium. This work was all because, the interest in these genes is part of the ENSA project, which now stands for Enabling Nutrient Symbioses in Agriculture, if anyone's interested. And I've just mentioned one paper actually talking about the role of RLK4, if anyone's interested in looking that up. So, yeah, so we looked at this and just put some data here. I would say in these experiments, actually, the golden promise was not good. But I think you can immediately see that we're getting shoots isolated. The shoots are almost always transformed. These numbers, 78, 64%, 42, 30, we're well above that 20, even that 25, if golden promise was behaving itself, that 25% level. So this is really transforming very, very well. And I think it's interesting that, and also alongside the study that I just showed you, it's an interesting area to look at for making further improvements in transformation, in barley, because I think there's a lot of opportunities here. So that I've talked about this first barrier, because the second barrier, and the one that's got a lot more attention, is the barrier to regeneration. And if you look at the literature recently, there are many options actually. A lot of work in wheat and also in other crops, not so much in barley, very little in barley actually. And here a range of different transcription factors and different so-called regeneration factors have been used to boost transformation efficiency. Let me just show you a little list of these and just run through. I don't like slides with lots of text, apologise for this, but just put it anyway. So a lot of the early work, baby boom bushel, it's now been shown to give a sort of 5 to 6 times increase in transformation efficiency in wheat. Those of you who listened, I think it's the last but one seminar in this series. Juan Demonardi gave a lovely seminar talking about the use of GIF system in wheat and how it's completely transformed wheat transformation. So this, gives very high efficiencies in wheat, and it enables transformation of a whole range of varieties. And I'll talk about where we are in barley in a moment. Then there's a wheat WOX5, another study that was published. Again, really high efficiencies over, in this case, 31 different wheat cultivars by overexpressing the wheat WOX5. So another opportunity. But I will say in both of these last two, there is an effect on phenotype. So that needs to be mitigated in some way with some system to overcome that problem. And then just the final one, this is a more recent study that I found really interesting, because this was, it actually identified 2 additional transcription factors, which if you overexpress them, they enhance transformation efficiency in different wheat varieties. But it looked at it in a slightly different way, because it really looked at uncovering these sort of regulatory networks that are underlying this capacity for regeneration. And this was really interesting, because there was a lot of difference between different genotypes. And so it almost looked like, if you understood this and you knew what was important for your particular variety of interest, you might be able to design a bespoke strategy to overcome the problems in that variety. So I think this, this is, again, a fascinating area that's moving very quickly. But then the question is, okay, but what about in barley? So there's lots of advances in wheat, but what about in barley? So I think Juan mentioned something about this in barley briefly, but we tried the wheat GRF4, GRF1, the wheat construct under a strong constitutive promoter in barley. This is what it looked like. It basically gave up at this stage. It didn't really regenerate compared to the control. So first attempt, that wasn't looking great. But, there are other opportunities. So I've mentioned WOX5. So we then had a look at WOX5. So again, what we're looking at here, this is a control, not such a good one. This is the wheat GRF4, GRF1 for comparison. You will see it produces loads of lovely looking callus, but regeneration is a problem, because it seems that it's just, if you like, it's too much in that diploid barley genome, whereas in hexaploid wheat, it copes with it. This is the wheat WOX5. So not great, some decent looking callus, but what Tom did, he isolated the barley WOX5, thinking actually maybe we'll try the barley version. And that looks a lot better. I don't know if you can see on the slide, but there's a lot more greening up, a lot more plants being produced here. So we've got GRF4, under a strong promoter, this is not, but clearly there are options to do something about this, to maybe use an inducible system or different promoter. And we've got the Barley WOX5, which is looking promising. So, that sort of gives us a couple of strategies in barley. So again, we're looking at some control plates here. I'm showing this is later stage, showing regeneration. Here we're looking at the GRF, GIF again, but under a different promoter. So this is the PLTP promoter. It's a phospholipid transfer protein promoter. It's actually expressed in the immature embryos, which is our target tissue, and also in callus. But the expression is limited in other tissues, so particularly those reproductive tissues where the problems of regeneration and sterility kick in at a later stage. So this is an option. And of course, there are also options for inducible systems. And this is something, again, that Tom has been looking at. I'm not talking more about today, but there are definitely options there. And this is the Barley WOX5, and you can see it's looking very good. Lots of regeneration. But the important thing is, what do the plants look like? And here's a picture just to show what the plants look like. And you see the control. So there is a phenotype with the WOX5. But we have seed. So we know we've got spikes, we've got seed back from it. And this is the GRF4 under the PLTP. So I think, the message here is that there are some good opportunities to really do something in barley about this problem of genotype dependence and boosting efficiency. But, it just needs a little bit more tweaking to get, the perfect system that's going to work ideally across the board in barley. I think when we first started looking at this, we assumed that everything that worked well in wheat would, of course, work in barley. And we, unfortunately, were disappointed that that was absolutely not the case. Okay, so that's enough on the delivery. So I think of course, there are a load of other options for delivery as well, new opportunities for delivering in different ways, which I'm not going to cover today. But I think it's an area that it is a bottleneck, but it's something that is now, I think, solvable, and there's real progress being made. So now to go on and just talk about the toolbox. So this is it's a Golden Gate based toolkit that Tom has developed. And as I said before, about 70 constructs. So Tom is just finishing off the paper, which I'm sure will make available at the first opportunity, and all of the vectors, all of the components will be made available in AdGene, and that also will be done very soon. So you're getting a bit of a sneak preview, but hopefully this will all be available quite quickly. So, to start describing the improvements that have been made, we need to go back to what our original system, our generation 1 system, if you like, looked like. And we did a lot of genome editing using this type of system, whereas for each target gene, we produced 2 constructs, usually with a pair of guides. And we produced about 20 plants for each one. And then we screened them and we identified edits, and then we went through to the next generation and found our homozygous plants in either the T1 or the T2. And okay, this worked pretty well. If you just look at the bottom here, this is a whole range of different target genes. Most of them successfully edited, some at very high efficiency, some low, but a lot of variability. And overall, for the average efficiency for single knockouts, 33%. And if we're doing more than one, 34%. But that, I think, is where we had already identified guides that we knew were functional, which is why that's perhaps a little bit higher. And of course, if you want to edit more than a single gene, then having to identify functional guides in a first step, again, it's a lot of time, and you don't really want to have to do an initial step to check everything out before going for your multiple knockouts. So basically, as this is one of Tom's slides on there is room for improvement. And another way of looking at this, which is quite important, is that the proportion of the plants that we produce that are actually edited. So this again is the old system. The edited ones are in orange. So you can see in a lot of cases we produced lots of plants, a very high number of plants, and only a few of them are edited. this is really wasteful of resources, because we've got a lot of plants with, it's a lot of tissue culture, a lot of work, and very few edited ones at the end. Although it's successful, again, a lot of room for improvement. So the first improvement, if you like, that I want to talk about, I'm going to trace this back to some really early work that was done a very long time ago, where we were looking at the impact of introns and looking at intron-mediated enhancement. And we found that actually by including introns in the coding region of a transgene, in this case the firefly luciferase gene, we could get big increases in expression of the gene. So it's not really a surprise that this similar idea has been used in genome editing as well. And the first example was from Jen Sheen's lab, where what they did was included a single potato intron in a plant codon optimized Cas9. And they showed that this, including this intron, gave very high editing efficiencies in Arabidopsis and tobacco. This was followed by a study from Sylvester Marinette's lab, where they went a step further. Rather than a single intron, they actually introduced 13 introns into a maize codon optimized Cas9. And this again gave a dramatic increase in editing efficiency in Arabidopsis and Benthamiana. If you just look at the knockout phenotype and just look at these final two, or the final three constructs here that contain the introns, you'll see that it's gone from nothing to these high percentages of plants with the knockout phenotype. So this was looking, this really made a big difference to but initially in Arabidopsis and tobacco or Benthamiana. So we're obviously interested to see if this would help us in barley. So what Tom did, he took 3 Cas9 variants. So the first one is the standard one that we used in our initial system. It's actually the original human optimized Cas9. The second one was the version, the plant optimized Cas9 with a single intron, and the third one was the version with 13 introns. And he used these to target 5 target genes, and each of those 5 genes was targeted with 4 individual guides, so a total of 15 constructs, and then had a look to see what the results looked like. And this shows what he got. So this is the five target genes, the 3 different Cas9 variants. So the standard one, the one with 1 intron, and you can see if we compare to these numbers in the standard, this is with the single intron, it's pushed it up 80, 90% or more. And then the one with 13 introns, you see for 3 of these target genes, it's right up at 100%, and the other 2 are quite high. So clearly both of these with the introns are working extremely well. If you look at the average sort of efficiency of mutagenesis over those 5 genes, it was 33% for the standard one, 88% for the one with 1 intron, and up to 96% for the one with 13 introns. Now, I'm mainly talking about barley, but obviously it's interesting to know what's happened, how this performs in wheat as well. So I just put in one little bit of data here in wheat. This is editing a single gene. It's actually, we're going back to the RLK4 that I mentioned before, using the best Cas9 with the 13 introns, and looking at editing in the 3 wheat genomes. And you can see that in each genome, this gene is edited at extremely high efficiency using this very efficient Cas9. So it also works really well in wheat. So obviously this informs Tom's decisions about including these improved nucleases in the toolbox. So now I just want to move on to Cas12a. Sometimes using an alternative to Cas9 is an advantage for various reasons. And Cas12a is particularly interesting and valuable. It has a number of features that that make it valuable, in addition to some of the IP considerations, which some people are particularly interested in. The PAM requirement is different. You have the NGG PAM, obviously, with Cas9, whereas Cas12a, that used to be called CPF1, has a T-rich PAM, which means you can use it in slightly different applications. It cuts away from the PAM site, and it leaves sticky ends as opposed to being a blunt cut with Cas9. And the types of deletions, the types of changes you get are different as well. So whereas with Cas9, you tend to get very small deletions or perhaps a single base insertion or a single base switch, with Cas12a, you get slightly larger deletions, and they often tend to be in multiples of 3. So, Tom was interested in looking at Cas12a as well. And he started off by taking 4 Cas12a variants that were available. So there's a human optimized one. There's a rice codon optimized one. There was the human one, which actually contains a temperature tolerant mutation. This is one that Tom actually added this mutation in. And there's the plant codon optimized one with the same mutation. And then what Tom did, he thought, well, the addition of introns was so effective in Cas9, it seemed to make perfect sense to try and do a similar thing with Cas12a. So that's exactly what he did. He's added 8 introns into Cas12a. So these are the different variants. This is just showing the constructs and these different variants again. So you can see them. You can see them here. As I said, the temperature tolerant mutation, that's the D156R mutation that's been introduced, and that's present in these last 3 constructs. The guide arrays are the same for all of them. And I will say a bit more about multiplex editing and the architecture of guide arrays in a minute. But basically it's the same. So what did we get from this initial set of constructs? Well, basically by far the most efficient one was the one with the 8 introns, which boosted the efficiency actually from, if you take this version, if you compare this one, the third one with the bottom one, it basically, this here it's not working at all, and it's been boosted up to 87%. The second best one was this, the human optimized Cas12a with the temperature tolerant mutation. It was sort of somewhat surprising that this first one didn't work, and I think that's something that Tom's looking into at the moment. So okay, so that was that's Cas12a. Also, because we wanted to see whether this would function well in wheat, so we had a look in wheat. At the same time as beginning to look at construct architecture. So here we're looking at the 2 Cas12a variants that work best in barley. So it's this first one without the introns, but with the temperature tolerant mutation. And these 2 both have got the 8 introns added. The difference between, so the difference in the guide architecture, the first 2 constructs, each guide has its own promoter, and it's flanked by these hammerhead and HDV ribosome sequences. In this third one, this is using a tRNA-based architecture. So it's just to give because again, in the literature, there's an indication that this actually might be quite valuable. And here's the results from that. Targeting 2 genes in wheat, so GW2, GW7. You can see the guides. We've got 2 guides for one and one for the other. If you look at the first construct, you can see the editing efficiencies over the 3 wheat genomes with the 2 different targets. Yes, not bad. So I think 29% up to 65. The second construct, the first one with the introns, these have been boosted up to a higher here of 88%. And in that final version 3 construct, if you like, which had the tRNA architecture, you can see it's even better, very high efficiencies across all of the genomes and both of those target genes. So this is giving information as to what might be best to include in a toolkit that's hopefully going to give the highest efficiency editing. So continuing, a little bit on guide architecture. This study was looking at that. So I think we had the idea that if you just used a guide architecture with a single promoter driving the guide array, then there seemed to be a tendency that you would get a lot more activity of the first guide than the subsequent guide. So we wanted to have a look at that. So that's what this version 1 construct is. It just has a single promoter driving the guide array. And this second version, again, each guide has its own promoter. We're using here the wheat U6 and the wheat U3. And is also flanked by these ribosome sequences. So in this study, there were 5 different genes targeted, 4 guides for each target gene. And basically the finding was that this second version, where each guide had its own promoter, it gave a higher overall efficiency of edited plants. But of course, the version 1 was more efficient in position, in the array position 1 for the position 1 guide. So you can see that if we look at this first target gene, VRS1, very high editing efficiency for the guide in position 1, a little bit for the second position, nothing for the other 2. Whereas the version 2 architecture, there's quite, much more even editing across all, and editing across all of those, using all of those guides. And we see a similar pattern for the other target genes as well. So again, this sort of informs the, depending on what you want and the outcome that you want, the sort of design that you might want to consider. I realized when I was putting this talk together that I hadn't actually put in any slides showing any edited plants. And because I mentioned VRS1 here, I've stuck in a slide showing a VRS1 edited barley, just for good measure. So if you knock out VRS1, single knockout in Golden Promise, which is a 2-row, you will create a 6-row Golden Promise. And it's a beautiful, and we saw this in the primary transgenic plant. So it's a nice phenotype to look at for edited barley, which is why I put it in. Okay, so just going back to Cas9 with the Introns, and just another comparison of the difference it makes having different guide architectures. So in this example, 3 genes targeted simultaneously, and there are 3 constructs with different architecture. Again, each target gene had 4 guides, so we've got a total of 12 in each of these constructs. The first architecture, again, each guide has its own promoter, either a wheat U6 or U3 or the barley U3. In the second construct, using a single, this is a rice ubiquitin promoter, driving the guide array, where all the guides are separated by ribosome sequences. And in the third construct, again, a single rice ubiquitin promoter driving the array. But in this case, the guides are separated by cis4 recognition sequences, and the cis4 protein is co-expressed, which you can see here. So this actually, I have to say, all of these worked very well. If we look at, so construct A, construct B, well, construct A would basically over those 3, with 100% editing over those 3 targets. So with each guide with its own promoter, absolutely super efficient. Very little reduction in this second architecture using those single promoter and those ribosome sequences flanking the guides. And still the cis4 system not as good, but it's still reasonable editing efficiencies there. So again, a lot more information for Tom to think about what to include in his toolkit. So I just want to mention briefly the idea of so multiplex. I've talked about multiple edits in a few of those examples, but because we realise we've got a really efficient Cas9 and a Cas12a, both containing introns. If you wanted to do something quite complex, some of your target genes might be more suitable for Cas9, and some of them might be more suitable for Cas12a target sites. So could you use both of them together? And so that was something Tom looked at. And he also really thought carefully about the promoters he was using and the position of the promoters, which I'm sure he'd say a bit more about if anyone wants to a bit more information on that. So yeah, this is what he looked at. Actually, could he include the most efficient Cas9 and the Cas12a in the same construct and use them together in a sort of multiplex editing system? And basically the answer was yes, you could very successfully. So again, this is the construct. You've got the Cas9 with 13 introns, you've got the 12a with 8 introns, both driven by major ubiquitin promoters. Driving two, here we've got two different arrays here. I haven't included all the detail because this is a bit complicated because there were 4 constructs. 2 of them targeted 2 genes, 1 3 genes and 1 4. But the outcome was basically that where Cas9 and Cas12a were used together, they edited all the target genes in all the constructs. So, and I've just taken the data from the construct with the targeting 4 genes, where target 1 was just targeted with Cas9 guides, and the other 3 target genes had two guides for Cas9 and 2 for 12a, and you'll see very high editing efficiency over all of the targets using that approach. So that just shows that it's possible to put these things together. Right, so just a couple of slides to sort of summarize the changes that we've made. So from the original system, what we called system 1, our original editing system, if we were doing double knockouts, really the efficiencies were very low. When we started making the improvements, they went up dramatically. And so even getting triple, and in this case, the quadruple mutant was super efficient. So yeah, this has made a really big difference. But perhaps another way of looking at this is just to make a comparison now. So this was I've shown you this before, this was the old system. And you can see this variation in the percentage of edited lines. And this is the new system. And if you actually look at the percentages, you can see the difference is really quite dramatic. So the old system for single gene knockouts are 33%, multi-genes 34%. The new system for single 91% and the multi-gene knockout 61% as an average. So a really big difference. But another important thing to look at is the proportion of the plants that are edited in the new system compared to the old one. Because rather than producing all these plants shown in blue bars that are not edited, they're wild type and just a few edited ones, you will see now that vast majority of the plants we produce are edited. So this is really quite important because it means that we can produce, so here, we're up to 60 plants, we're making a lot of plants, a lot of work. Now here we've got much lower numbers of plants. We don't need very many. So we can really reduce the amount of tissue culture. We can reduce the glasshouse space that we need, which is expensive. Much easier genetics, because it just makes life a lot easier. And we can tackle more challenging projects as well, because because you have the efficiency to have the confidence to do things that are more difficult. So this is just showing the difference that these new tools have made. Okay. So I've talked about mainly sort of targeted mutations and multiple knockouts. I've talked about the nuclease variants. I haven't had a chance to talk about anything else. Tom is also very interested in gene targeting, and there's a paper you can have a look at on that. And now using some of the new tools to improve gene targeting, and I think this is going to be important going forward as well. So I'm excited to see what happens there. But just keeping an eye on the time, we're okay, I think. Not too bad. Want to say a few words before sort of opening up for questions on the regulatory status, because it's all very well as having this, these fantastic tools in our hands, and we can do all these things, and we could produce some exciting material that we think may have a real impact if it could be, applied and used commercially. Then we need the regulatory system to allow that. And this is slightly out of date. This was put together by Penny Hundleby, the map showing the situation worldwide, where it's basically everything in green. The simple gene edited plants are regulated in a very similar way to conventional plants. There are areas under review, which at this point it did include England, and in red, Europe in particular, the plant's still treated the same as GM. So, but there's been a huge amount of activity in this, especially in England, say recently in this area. So we have the Genetic Technology or Precision Breeding Act, which has been right through Parliament, has received royal assent. And this bill basically it makes provision for the release and the marketing and risk assessment related of precision bred plants and animals. So it's all to do with, it's considering those two areas really. So firstly, release, which is research material, and then taking things forward for marketing to use them as food and feed. And the main, if you like, the main things in the bill listed here, the idea was to remove plants and animals produced through precision breeding techniques, including these simple genome edits, from the regulatory requirements that would have been applicable had they been considered GMOs. There was a requirement for a suggestion to have notification systems, so one for the research notifications and one for marketing purposes. There was a requirement to take pay particular attention to precision bred animals to ensure animal welfare, but to also establish a science-based authorization process for food and feed products developed using these technologies. And that's where, okay, so first of all, just thinking about the release. I mean, many of you will be familiar with this system because I think there may be people in the audience who have done this, have edited material, which has gone through to field trials, following the notification that their material is a qualifying higher plant. And it's clear that qualifying higher plants can be developed using genome editing techniques such as CRISPR-Cas9, as long as they don't contain foreign DNA, and they could have been produced using other methods. And this material, this has really opened the door for looking at the performance of some of these plants in the field, which is a huge step forward. But the other thing that I did want to mention, because the next step is not just field trials, it's actually marketing food and feed derived from precision bred organisms. And this is where the Food Standards Agency comes in. And the Food Standards Agency they actually commissioned one of their advisory committees, which is the Advisory Committee on Novel Foods and Processes, ACNFP, which I will admit that I sit on, to basically provide some information towards informing the approach to the management of the safety risk of using PBOs in food and feed. And so the committee actually said that they've seen no evidence that precision bred organisms are intrinsically more hazardous than traditionally bred organisms, which I think is completely fair. But it is recognized that there's this new technology, there's a range of outcomes possible, and it's very rapidly developing. So this may justify some additional scrutiny to ensure that risks are identified and effectively managed. So what the ACNFP committee did, they actually developed two potential models. It was decided actually at quite an early stage and agreed that a two-tier system for this would be quite sensible, where precision bred organisms that fell into tier 1 would be very similar to traditionally bred varieties that consumers have already consumed and they're familiar with, and safety risks are understood. And there should be a very simple route to market. Things that fell into tier 2, this is where maybe the risks were not fully understood. So it could be because the plant was novel, it could be something that consumers were not familiar with, or it could have compositional changes that could perhaps be expected to affect toxicity or allergenicity or other safety concerns that would need to be looked at in a little bit more detail. So basically the big question was, okay, but in making the decision as to whether things are tier 1 or tier 2, what data might be needed? And this is where ACNFP came up with two models. And basically, I don't need to read this, but basically model 1 has very limited data, compositional data required. And it's just to, if you like, verify that the change that's been made really is what you say it is. And in model 2, there's a requirement for additional compositional data that will be considered up front to capture maybe any wider consequences of the change that's been made. Now, if just very recently, there is a paper that you can go online to here, have a look at. If you're interested in this area, I'd recommend go read the latest updated version, which is a paper that's now going to the FSA board with some recommendations based on all the information that's come from the committee, and I've just picked out a couple of recommendations here that they're saying, taking all factors into account, they recommend the two-tier regulatory approach. It's fair enough, that's been agreed, but using the data requirements in ACNFP Model 1, which remember is the lighter touch. So that's a recommendation. And a second recommendation I just put in here, they recommend that industry has legal responsibility for undertaking the initial triage, determining whether a PBO is tier 1 or tier 2, but there would be a notification system necessary. So it's going to be, it's going to be very interesting to see what comes out of what is actually agreed and what comes out of this, from this document, what is agreed or not agreed. We don't know yet, but it's a fast-moving and interesting time. But I think you can say that things are. Yeah, I think it's very encouraging. And just to say, we're talking about the changes obviously in England, but things are moving in the EU as well. There's a proposal that's being put forward to update the EU rules and basically to again mean that certain the GMO regulations will no longer apply to plants created using techniques such as CRISPR, as long as those are those simple modifications that could have been achieved using conventional breeding. And again, if anyone's interested in having a look at that. So I think I have probably talked for long enough. So all I need to do is to thank, and this is actually an old slide, some current and old previous members of the group, but particularly to thank Tom. It's mainly his work that I'm talking about today. Other people I need to thank, Alison, who's retired recently, who's carried out most of our barley transformation over many years. Now Macarena has taken on that role, is doing a fantastic job. Martha, who's helped Tom a lot with the work that I've been describing today. Kaylee, who's also helped out, has done a fantastic job of caring for all our plants and keeping everything ticking over, and everyone else who's contributed in whatever way. So I will stop there and I'll stop sharing if that's all right.
Fantastic. Thanks so much. You're getting some claps in the. I don't know if you can see the participant windows now, but you're getting a whole bunch of
Thank you.
claps. So, Wendy, we've got some questions in the chat. We've also got some people with their hand up. So why don't I start with people that have got their hands up? So Christoph, you had your hand up first, I think.
Try to clap, Robbie. Very nice presentation. Thank you, Wendy.
Well, listen, while people are thinking in that case, I'll ask some of the questions. And well, I'll ask Wilma for a start to ask the question that she's put in the chat. Do you want to do that, Wilma?
Yes, of course. Wendy, first of all, thanks for a great presentation. One thing I was wondering when I looked at this high editing efficiency of the Cas9, the Intron optimized, and also the Cas12 Intron optimized, is if this is like at the cost of having more off targets, or is it still very specific?
Okay. So, I don't know if Tom will want to comment on that. I mean, we're quite careful with our designs to avoid off targets. And we haven't. I have to say, we haven't looked in a lot of detail. So I don't think I can give you an absolute answer to that. But I wouldn't expect it to increase the off targets a lot. I mean, I think I didn't say, but clearly in most of our experiments, once we have our edited plants and in the T1 generation, we've found the homozygous or biallelic edits that we want to keep. and we've identified the homozygous lines, then, and we will segregate away then the Cas9 and the guides. So we're fixed from that point. There'll be no further editing. Clearly, if the Cas9 was left in, you might then worry. You've got a very active Cas9 that's left in, then I think you might have a concern. Yeah, but I don't know. Yeah, I don't know if Tom is around, and if he wants to come in on any of these questions, Tom, please do.
While Tom's thinking,
Yeah.
there's another couple of questions in the chat.
Okay, thank you.
And Crispus, I see, has his hand up. So let's do one from the chat first. I had a very simple question for you, Wendy, and that is, you mentioned the not off-target effects, just the phenotypic effects of WOX5 transgenics. I presume they disappear as soon as you get rid of the transgene. Is that correct?
I would assume so, yes. Now, I don't think we've taken those through to the next generation and grown up the next generation yet.
All right.
So I couldn't say for sure, but if we had a system, if we were able to segregate away the transgene, then you're right, it shouldn't be a problem, because it's similar to if we were using one of the other systems. If we can then, once it's segregated away, shouldn't be a problem for the next generation. Yeah.
I'm going to keep the stage just for a minute, because I can't remember if you showed data about the impact of varying the number of introns. And I wondered, I mean, really, why do you think so many introns boosts the editing frequency so much? I mean, why not one intron or two introns?
Yeah, that's a really good question, and it is something we're looking at. Again, I'm going to ask Tom if he wants to just say what we're doing on that at the moment.
Craig Simpson might comment as well, but there you go.
I'm not sure if, because I can't see who's on the call. So it is something, it is a good question. I'm just having a look at, so I'm not picking up on messages and things here. So it is something we're looking at, because Craig's saying, have lots of questions about that. He might want to make a comment as well. I mean, it is something, because the question was, okay, do we really need all those introns, exactly as you say? And it is something we're looking at. So what Tom is doing is looking at this and saying, okay, maybe if we cut them down, so maybe we just need the first intron, or the first few, or the middle few, or the end few. And he has actually split them up, and we are looking at that, which will give us an indication of whether actually they're all necessary, or we've just followed the we used this what Tom did. He used exactly the same software and the same system for deciding on the introns, the intron positions, as was used for the Cas9. I don't know if Craig wants to come in on that as well.
He's got lots of questions, apparently. Do you want to answer them, Craig, or ask them, or?
Yeah, I can ask a few questions. Hi, Wendy. Thank you very much for that.
Hello.
It was very refreshing to see so much molecular work being done. You don't see many talks like that anymore, so it was really nice. I'm obviously interested in this the intron part, because it's fairly well known that introns boost gene expression. So some of my questions were, do the introns splice? Have you looked at that? Do you get an induction of expression of your Cas9 in with these introns? And then the position of the introns is usually quite important towards the 5' end as well, for induction of high levels of expression. So I was just kind of interested in all these sort of questions.
Yeah, and I think the answer is that as I said before, this is something that Tom is looking at, and I was hoping he might be able to pop in and up with it, but I don't know if he's actually able to connect in at the moment in thought, but never mind. So in terms of the position, I mean, I agree with you. What we've seen from that very early work that I mentioned on one of the first intron slide is that it's always the first intron that's the most important. So that's sort of what we would expect. Yeah. But we don't, in this case, we don't know. So we're having a look. So we haven't got that data yet, but hopefully it's sort of what I would expect. But sometimes we're surprised. So I don't know.
Yeah. So that's what I would
Um,
expect as well. And
Yeah, it's what you would expect as well. So that's good to hear. Splicing again, I don't know. You know, I think there's an awful, if we had, you know, lots of time and someone to sort of really dig into the details of this, there's lots of things that we could do. But unfortunately we don't. I mean, I guess our aim is once we've got something which is just working super efficiently, then the temptation is we don't really need to dig too much. You know, we can just use it. So it's finding that balance. Yeah, but maybe an interesting student project, maybe.
yeah, potentially,
Yeah.
Wendy, we've got another couple of questions in the talk that I'm just going to read out because in particular, Lalita's put in a question twice. First of all, the comment was, very nice talk. So I think we all agree that, very nice talk. Did any helper plasmid, was any helper plasmid used to improve efficiency in barley?
No, okay. So I'm not quite sure what it means by yeah, helper plasmid there. I think the answer is no. This is the so a lot of the original constructs, this is the sort of pea green, pea soup system. And you might be thinking about the pea soup variants with perhaps the ones that are used in the wheat system with additional virulence genes. We don't use them in barley.
Okay.
So I think that we have had a look in the past. I don't think we feel that there's an advantage there. So from that point of view, no helper plasmid specifically
Okay.
to boost.
Now we've got a political one from Nick Pitts. How does the development of the potential commercialization of PBO in England impact other nations within the UK? For example, Scotland.
Yeah, good question, and I'm not sure I'm the best person to answer that. I mean, clearly the changes in, you know, the bill only applies to England. It doesn't apply to Scotland. But yeah, I mean, it's a good question because, you know, if the regulations are different in England and products are available on the shelf, then presumably they could be available in Scotland as well,
Absolutely.
but you wouldn't be growing them, I guess.
I presume we'll follow the European Union, which is what we like to do.
Yeah. Okay.
Okay, so what other barley cultivars were you able to transform successfully other than Golden Promise? What ones have you tried?
Okay, right, now I have to think back. So in the past, we've tried a few quite unsuccessfully. We have a mutant line that transforms very well, and we still haven't quite got to the bottom of why that transforms very well. There's some work that we're doing with Kay Trafford. And, we've recently tried, there was interest in Bowman and Morex. We haven't gone back to Morex again. Morex was difficult. Bowman, we've got a few transgenics. In the past, we've looked at Optic. Again, we couldn't transform it, but I would like to go back and have another look at that again. But I think we're at the stage at the moment where we just need to really optimize, you know, have the data, really optimize our system, what we're going to go for, and then try it over a panel of different genotypes.
Okay.
So I think that's what needs to be done next.
That's good and makes sense. Listen, there's just a couple of comments I want to make before we let Wendy go and thank her again. First of all, Barno, if you go to the link that I've pasted into the chat box, it'll take you to all of the previous seminars, and Wendy's will be on there in a few days' time. There was one other comment I wanted to make. No, I think that's, oh, no, it wasn't, sorry, there was one other comment I wanted to make, and that was whether or not, where is he going? I was going to ask the person that had their hand up if they wanted to ask a question, but they're gone. Okay, no problem. So listen, Wendy, all that's left for me to say is thanks very much. That was great. And like Craig said, it was nice to see some gene jockey stuff happening again, just to let us know exactly what's happening in the real world. So I think if everybody can just clap their hands virtually, we'll thank Wendy, and yeah, look forward to the next seminar as well. Thank you, Wendy. Thanks very much.
Thank you. Thank you.
Oh, there.